you so much for having me. I am really excited to talk about what I did a lot for my thesis, and I love answering questions. So I have some set places where I think are natural breaks for questions, but please, please, please ask questions. So paleontology, what do we do? We do a few things. We mostly collect stuff when we answer questions. So just a quick recap. There are super, super cool animals that lived in the past. This is one of them that is near and dear to my heart. This is Dreadnoughtus strani, a dinosaur I got to work on. And hopefully they die next to some body of water that allows their bones to become fossils. And then people go and collect these fossils. And I think we might see something about that next week. So then we paleontologists go grab those fossils, clean them, prepare them, and put them in super cool museums like the museum in Virginia and my home museum, the Academy of Natural Sciences of Drexel University. What do we do once we have those bones? So I'm a paleobiologist, and what that means is I love thinking about these animals as if they were alive. So look at all these pictures. What do we see? There are some animals swimming, lounging, fighting, running, growling. These are the sort of questions that I'm really interested in. What were these animals like when they were alive? So my job is pretty cool. I get to recreate fossils for a living. So that's today what I'm going to talk about is sort of this process of answering questions in paleontology, experimental paleontology, and how do we get motion from bone? Here's sort of a general guide to answering questions in paleontology. We have to find, once you have your question, we have to find the right animals, which involves a time and a place. So if you have questions about T-Rex, you shouldn't go look for T-Rex in the Permian. If you have questions about swimming, we shouldn't look at terrestrial animals, so animals on land. Once you figure out your question and the right animals for your question, then we need to think about the body part you're really interested in. Do you like toes? Do you like tails? Do you like teeth? Do you like eyeballs? Eyes might be kind of tricky because eyeballs don't fossilize. And once we have our animal and our body part of interest, we need to think about our experimental design. What are we actually going to do with these taxa? So I'm going to walk you guys through what I did for my, my thesis. So my big question, I love spines. I love the spinal column. I love vertebrae, and we're going to talk about those parts. So that's my question. The animals I'm interested in, right, the right time and place, are Permian tetrapods. So tetrapod means 2 arms, 2 legs. That's us, that's giraffes, that's snakes are tricky. They are tetrapods, they just lost their limbs. Most animals you can think of that aren't fish, that are on land and have a spine on the inside, so not bugs, are your tetrapods. So the body part that I'm really interested in, like I said earlier, spines, vertebrae. I don't like limbs, I don't like noggins, you know, your head. And then experimental design. So modeling, 3D printing, the stuff we're really going to talk about today. So spines in ancient animals. Here we have a salamander, right? You can see he's walking, sort of twisting his body side to side. And then watch this. When he swims, it's all side to side spine stuff, right? So while there's a lot of debate as to who was on land first, how did animals run, if they ran, there's a pretty solid consensus in paleontology that ancient animals used a lot of their spine, right? So think of how like your salamanders walk, right? So spines are made up of vertebrae. So if everyone takes their thinking finger and puts it on the back of their neck towards your shoulders, you feel that lump? Yeah, so that's the top of your vertebrae. That's the top of C7. So you can feel part of your vertebrae down your spine. So there are a lot of different vertebral types, but a really common one now is what we call monospinalous. When I say monospinalous, you can just think whole vertebrae. And these whole vertebrae are made up of a neural arch, right, which is sort of that tip you were feeling right there. Let me grab my laser pointer. So when you put your thinking finger on the back of your neck, you were feeling this part of your spine, which is super cool. There is a centrum, right? It was sort of this weight element, this weight-bearing element, which you will not feel ever, I hope, in your own body. And then there's the spinal canal, right? So how our brain transmits information to the rest of our body is that the brain extends through this canal called the spinal canal. And this is in almost every vertebrae you can think of today. So human, dog, crocodile, snake, cat, almost everything, including, again, this very cool dinosaur that I got to work on, Dreadnoughtus. Very silly picture of me. But let's compare that to the fossil taxa that I work on. So again, here is this silly, get out of here, laser pointer. Here is this picture of Dreadnoughtus. That's one of its cervical vertebrae. So again, one of those neck bones. And it has, so let's compare it to a multi-part vertebrae, which is one of the past. These elements are sort of similar. There is this neural arch, again, that top part of the vert. There is this centrum, right to the bottom part of the vert. Then there's this part sort of in the middle, the spinal canal that goes straight through. But what the heck is this thing now? We have this new element. This is new. And this we don't see in dogs or crocodiles or snakes or dinosaurs or almost every animal we can think of today. And those are the vertebrae that I'm interested in, these many part vertebrae. And there were a lot of them in the past. So my work is in the Permian, which is about 100 million years, give or take, before the dinosaurs. And there were tons of different variations of vertebrae in the past. So again, we have these neural arches, and then there's that centrum that I talked about earlier. But again, we have these new parts, and what do? They look a little different. We don't see them that often. So that's sort of my question. So these are the animals that I work with. So remember, in our procedure, we have our question, then we have to find the right animals in time. So my animals in time are Permian tetrapod, these super cool critters that are 100 million-ish years older than the dinosaurs. There's Archeria crassidisca. He's a swimming guy, so I gave him little bubbles. There's Diplocallis, right? So his head is shaped like a boomerang, very cool critter. And I think you guys saw that on the page for the talk today. He's also aquatic, so he also got some bubbles. Then there's Cacops, which is a terrestrial guy, and Diadectes. And these are all to scale. So Diadectes is this super big, I think like 2-meter long animal. He has a big old pot belly for fermenting vegetables because veggies are really hard to eat. And there are a lot of different vertebrae. So Archeria has one type of the many part vertebrae. Cacops and Diadectes have a different part. So Diadectes down here, Cacops down here. Diplocallis looks like it's all fused up, except it has an extra hole. Remember I said there's the one canal where your brain goes through called the spinal canal? There's another canal called the nodal canal that we don't have. So he's monospinalist, but there is an extra hole in it that's sort of strange. So, okay, we have our taxa, we have our time, we have the body parts we're interested in. How am I supposed to get motion from bones? Well, we can use some pretty clever techniques. So on the left, we have, the hand of a whale, dolphin, something like that. And let's say on the right, I had never seen these bones before. Let's say I'm a paleontologist. I went out to Kansas. I found these bones and I had never seen them before. Well, these look sort of similar. There's, a bunch of small bones here. There's a bunch of small ones here. Okay. There's 2 bones here. Okay. And these are 2 bones here. These, digits look like they could form some sort of a paddle. These digits, okay, maybe there's a paddle there. And what did you know? This is from a whale, and this is from a mosasaur. They're not the same animal, but their bones are similar enough in shape that I can make some estimates about how the bones moved, which is great. And if we have bones, we can then infer something about their motion, right? So if you have a bone that's sort of like a ball, and you have sort of a cup, like a ball and socket, right? Like our shoulders, we can estimate that, the bone moves something like this. If you have a bone that's sort of a cup and another little cup that sits inside, well, that's sort of like our elbow, right? So we have this door hinge joint, and that's great. So what this tells me is, okay, I have my vertebrae, I have my animals that I really like. If I have their bone shape and I can see those shapes in modern animals, then maybe I can see shapes, maybe I can understand how they moved. But the bones that I work on don't exist anymore. There's no animal on the face of the planet that has the bones that I work on. And that's kind of tricky. Well, here's another way to think about motion in a related way, are these ideas of living descendants, right? So there's a super cool T-Rex exhibit I just heard about. And we know that birds are living descendants of dinosaurs, right? Let's look at these 2 feet. We see that T-Rex, has 3 toes that all touch the ground. This is the foot from a cassowary. Also have 3 toes that touch the ground. Okay, that's sort of interesting. There's claws here. So what having a living descendant means, if I learn something about the living critter, so the cassowary in this case, or ostriches, or pigeons, or kiwis, it's a good chance that I've also learned something about the past taxon. That's great. So even if I don't have the bones, maybe I have a living descendant that I can work from. Another example of living descendants are mastodons and mammoths, right? So again, let's look at these two critters. We have, some tusks. Okay, that's a similarity. There's really big, what we would call columnar limbs. They look like columns. Okay, there looks like a big old part of the skull for chewing muscles. So if I learned something about the elephant, there's a good chance I've learned something about the mastodon. And this happens a lot in taxa that just went extinct. So pull of the recent. So animals that went extinct closer to today, there's more information on. But this is still really tricky for me. So my taxa don't have any bones like the ones that I can see today. So I don't have any bones I can go grab. And my taxa have no living descendants. So I can't go grab one of the living descendants and try and estimate the motion. And my taxa went extinct a really long time ago, 100 million years before the dinosaurs, right? So if all of geologic time is a calendar year, right? My animals went extinct sometime around Thanksgiving. The dinosaurs went extinct sometime around December 25th. So that's a lot of time that I don't have a lot of information on. And that's where I want to use 3D printing. Just to recap, my taxa don't have any living descendants, and their vertebral elements, those bones, don't exist anywhere in any animal anymore. And that's where 3D printing comes into play. So my cohort and I, Aaron Johnson, wrote a paper about 3D printing in experimental paleontology. And we wrote that because 3D printing allows us to recreate, actuate, and modify ancient life. I get to see my taxa again because I don't have any living descendants and because I don't have any bones. The best I can do is try to recreate the animal as mechanically accurate as possible and then figure out some information about them. 3D printers vary in a lot of mechanisms and capabilities. So a lot of people, if you've seen a 3D printer, might have seen something like this, which is your standard SLA sort of plastic printer. Another printer type are these resin printers, which is what I get to work on, which is super cool. There's like a vat of resin, and then the printing bed goes down. And it gets shot with a bunch of lasers, which is super cool to harden up the print. And then there are powder prints, right? So your gypsum powders and stuff like that. So we have our printer, and 3D printing is sort of a really fun choose your own adventure. So a lot of steps go into it. First, we have to create the model. And we'll talk about how we do that. We have to decide the material and if it needs to be hard, if it needs to be squishy. If I'm going to throw my 3D print into a pool, I need to think about being water resistant. There's a lot of pre-processing. So I have my 3D printing file. Do I want to cut it in half? Do I want to make a lot of them? The fun part, of course, is printing. And then that's not the last step, though. There's the post-process. So I have my 3D print. Do I need to sand it down? Do I need to cut parts off of it? And then my favorite part, experimenting. So let's go through these steps together. And then I will go through steps with my own work to give you guys all an example. So getting a model, there's a lot of different ways you can get a model. Surface scanning is one. So I don't know if everyone here has seen a laser scanner. Photogrammetry is a really cool way to get laser scanning. You can do photogrammetry with your cell phone. So if you want to get a really cool 3D scan of something, you should look at photogrammetry and just do it with your cell phone. And this is super useful for fossils that can't be moved. So really big fossils that are on plates, we can use surface scans to get some information, and that helps us with motion studies. So again, how animals moved in, that's sort of my thing. There's internal scans. So I don't know if everyone has seen like a CT or an MRI. They're in a lot of hospitals. So if you've ever like broken a bone or something like that, you might have gotten an X-ray, and that's what we use to get fossils too. And then there's computer generated models. So bones that don't exist, fossils that don't exist, I'm just going to make some, which is super fun and super cool, or some combination. Then we have material selection. So how much does it weigh? How much does it cost? Does it need to be squishy? Does it need to be hard? Things like that. Can the printer do what I want it to do? And some examples are plastics, right? So if you have a 3D print in your hand, you'll probably have a piece of plastic in your hand, powder, or the resins. And that's sort of what all these materials look like. And then when we have all these materials, we need to choose our, we're going to choose the material. So if I just need the shape of something, well, then I can use plastic. It's super cheap, super easy to use. If the material properties need to be brittle, then maybe I use a powder print. If I need to have flexible or different or high resolution printing, I can use a resin printer, like this study that was looking at dinosaur feet. Then there's pre-processing. So, okay, I've chosen my material. I want it to be stiff. I need to fix the files. I need to think about how I'm going to print it. So when you 3D print something like a Lego, right, do you want the world sitting down? Do you want the box sitting up? A lot of that's really tricky and a lot of practice. Do I want hollow or solid 3D printing? So if you have your 3D print in your hand, you can make it hollow. That's super cheap and saves material. But if you're worried about how heavy something is for your experiment, you don't want to make it hollow. You want to make it solid. And then, of course, printing supports. Sometimes 3D printers need a lot more support. So you can see here, this doesn't look right. And you're right, it's not right. This is a failed print because the printing supports weren't sturdy enough. So now finally we have super cool parts. So this is a lab that I worked in for grad school. And here's some prints that we worked on. And this is that resin printer. So here's the resin in the bottom. The lasers are down here. And once we have our 3D prints, we need to take care of them. We wash them with isopropyl, and then we bake them. We call this our air fryer. It bakes our 3D prints at like 450 degrees for an hour, and that changes the material properties. There's the print. There's all this post processing. So this is the work that I did. Here are some fresh 3D prints here. You can see they're still a little wet, so we have to wait for them to dry. This is what they look like when they're dry. And you can see these shears here. Well, these shears are for all these supports that I then have to cut off. So this is an example of my work. Finally, once we have our 3D prints, then we get to do our experiment. So we get to crush, we get to move, we get to put into different machines when we finally have our 3D prints. So before I go into my example of 3D prints, are there any questions?
If you don't have all the bones from an animal, how do you learn about it?
Oh, that's a really good question. So if we don't have all the bones from an animal, we'll go look to its nearest fossil relative and see if we can grab some of their bones. So for example, I work on these animals called temnospondyls. They're super old amphibians. And sometimes they don't fossilize all that well. And I'm missing some parts. So what I'll go look at is sort of a cousin of the temnospondyls and try and guesstimate and see if their bones might fit these other bones. There are some bones you can do that it's safer to do that with. So for example, if you have limbs, right? Limbs don't change that much. If you're super lucky and you have limbs of a left side, you can mirror them and have your right. So if you only saw my left hand, right, you could mirror this and then you have my right. So if you're super lucky, you have a left part and you can mirror it. Otherwise, you have to go look at the cousin of an animal and hope there's some elements there.
So for folks who happen to have 3D printers, have access to 3D printers, are these out there in digital libraries anywhere that people could access them?
Yeah, so there are a few digital libraries that exist. iDigimorph, I think, has a 3D printing library. 3M, which is like Morpho Museum something. So 3M has a few 3D prints. The Smithsonian recently has digitized a lot of its collections. So there are some pretty cool, I think there's a mastodon assemblage that the Smithsonian has digitized that's available. A lot of scientific papers might actually have some of their STLs up, though I haven't seen that too common. But if you Google cyber taxonomy, I think you should find some pretty cool STLs. iDig Bio, I think, is another one.
So is there any way to replicate the internal structures in the bones?
Yeah, so there's a slide where I show what a CT scan actually looks like. And inside of there is the inside parts of a bone. So the inside parts of some bones have this sort of spongy texture. And there are really cool studies that are trying to 3D print those structures and then crush them and see if they behave like we expect bones to. So yes, whoever just asked that question, you'll see a picture of what the internal structure looks like. And I didn't 3D print those elements, but you can.
Okay, thanks, Dr. Carter.
Yeah, okay, so let's keep going. So first things first, if I want to do my experiment, I need to get some usable fossils, right? Some usable files, your 3D printing files, your STLs. So what I did is I used a CT scanner, and these are my fossil taxa. So these are amphibians and, the animals I talked about earlier, about 250 million years old, on a dog bed for CT scanning. And once I have my CT scanning, I have to do something called segmenting, right? So we need to get the fossils out of the file. And it's sort of like digital fossil prep. So when I was younger, I actually did fossil preparation, which is a lot of scraping and gluing together. And that's sort of what we're doing digitally. And it's a little bit harder on fossil taxa. So this is an X-ray of a dog. And the soft part, so, these areas of black where you're not seeing anything, are a lot less dense than the bone around them. So bone, super duper hard, soft stuff, not so hard, right? That's why it's called soft tissues. But when you have a fossil, right, and fossils are permineralized bone, right? So it's like a rock that's sort of a fossil, instead of a bone, I guess I should say. The rocks surrounding the fossil and the fossil itself are really both hard. So the densities are similar. So it's really tricky to get them back out. So this is a fossil that I worked on, Diadectes. And what I wanted to get rid of, and you can barely even see the difference, are the spaces in here. So here is my fossil, right? This is a fossil vert. I see a 3D printing chat. And here is the material that I want to get rid of. And you're like, the color's really similar. Like, yeah, that's what's really hard about this process. So here's what a CT scan looks like on my screen. So we can see that same element, that same bone here in yellow. And I've started removing some of those processes, some of the sediment there. And you can see here's internal structures, right? So whoever asked that question about internal structures, I just said I only want, the outline of this element to be 3D printed, but you could 3D print these different cavities and internal structures. So this is how I got my elements to 3D print. So with that, we have our Echidnas taxa, right? So Archeria and Paraxis, K-cups, and our monospondylus taxa. So Diplocaulus and Diadectes. Remember that from earlier. So great, I have my STL files, but that's not the only step. I need to make some changes for my experiment, which is sort of the model manipulation part of 3D printing. So first I chose my material. I chose draft print, which is just a super fast plastic 3D print because I don't need, high quality stuff. I just need the shape. I made a flexible spinal cord and a flexible notochord where I needed, and a screw because I'm going to pull on this really hard, and we'll talk about that in a minute, and then a screw in the back because I'm actually need to hold to the table. So this is what those prints look like again. So we have wet, fresh out of the printer, dry, and then starting to take them apart. And I got to print with super cool rubber stuff. So this is a flexible resin that actually bends a lot. So this is what the model looked like, right? So this draft print, these flexible elements, and this is what it looks like in real life. And, you can see sort of these ridges here, but that's okay because I wasn't worried about the smoothness of the print. I just needed the prints. So let's go through these steps real quick. I have my fossil, that's Diplocaulus, like we've seen it a few times now. I have my CT scan. And once I finish segmenting out the CT, then I have my STL file. And this is a good example of another question that was asked earlier. What if you don't have all the bones? Sometimes you have the bones and they're just not great. So this element here is, it's crooked, right? A part of it is broken off here. So what I did in this case is I mirrored both sides. Once I have my STL, I can make some estimates using CAD or computer-aided design to estimate, make some hypotheses about how much I think this will bend. And then I can 3D print the elements and actually run my experiment. So quick, any questions about the process of 3D printing?
So when you've got those busted shapes, when the fossil's damaged, what are the different ways that you can kind of come up with what the bone would have looked like in the undamaged state?
Yeah, so there are a lot of different ways to do what's called retrodeformation. So let's get the bone back to the way it was in life. I'm lucky in that I can assume what we call bilateral symmetry, which means the left and right sides should look the same. So in my case, in something like Mesh Mixer, which is a free AutoCAD Mesh Mixer, which is a free software, I can take my 3D printing file, I can slice it down the middle and mirror it. I'm lucky that way. Limbs are very similar. But then if you have an element that's cracked and shifted and broken, there are a lot of different ways that people retrodeform those. So like swelling up a bone to its original size is something that some people do. Making estimates on other animals. So if, for example, you have, let's say like Velociraptor mongoliensis, right? And you have another Velociraptor that's not that species, and say, okay, well, they're both the same genus, so let me just copy paste bones from one Velociraptor to another species of Velociraptor. So we have our 3D prints. Let's get to our experiment. So here's some hypotheses. We have on the x-axis angular displacement. So that's just how far something's going to rotate around. And on the y-axis, a moment. So think about how hard I have to push or pull on something. In this experiment case is pull. So if I pull on something really, really, really hard and it doesn't rotate that much, we would say that thing has a high stiffness, right? It's really, really stiff. And if you sit in computer chairs all day, you're probably feeling stiff too. If I pull on something just a little bit and it goes really, really far, that has a very low stiffness. So those are the measurements that I'm really interested in taking for my taxa. And my hypothesis is those whole vertebrae, the single vertebrae, the monospinalist vertebrae, again, the vertebrae like you, me, a cat, a dog, a snake, a stegosaurus, I think they're going to be very stiff, that I need to pull on those really hard to get them to move really far. The rachitymous types, so these multi-part types, I think they're going to be very low in stiffness. I don't have to pull them that hard to get them to move really far. Okay, so I looked at 3 different directions. So there's something called axial rotation. So just imagine you're doing the twist. That's axial rotation. Side-to-side bending is lateral bending. So, you know, a different dance. And then dorsal ventral flexion, which is like stretching out your back and then curling into your tummy. So those are the 3 directions that I looked at. And let's look at some results. Okay, well, in doing the twist, our 2 aquatic taxa, Archeria and Diplocaulus, well, they were, there we go. Well, these guys were all equal, and K-cups and Diadectes were all equal, and that's not exactly what I expected. I thought that, you know, that Diplocaulus and Diadectes would be way stiffer, and K-cups and Archeria would be less stiff, and that doesn't look like that's happened. Let's look at side-to-side bending. Well, Diplocaulus had the highest stiffness, and then Diadectes, and then K-cups and Archeria were tied, and that's sort of what I expected, that Archeria and K-cups would be similar, but Diplocaulus and Diadectes I thought would be together. Okay, what about bending all the way back? Well, Diplocaulus was, you know, he bent the most, followed by Diadectes, and okay, that's kind of close to what I thought, but K-cups and Archeria being different, that's, If you're thinking, wow, that's all over the place, and none of that is what we thought, because we thought Diadectes and Diplocaulus would be together and K-cups and Archeria would be together. Yeah, that's right. None of it is what we thought it would be, and that's okay. That's hypothesis. Part of experimenting and doing experiments is being wrong, and that's okay. So yeah, we thought that those 2 would go together. They didn't. Thought those 2 would go together. They didn't. Whoa, double clicked. So yeah, all of my hypotheses were wrong, and that's okay. A lot of what I do in designing these experiments, a lot of what I do in designing these experiments is failing. I make mistakes. Couldn't get that last video to play. And if you've gone through this talk and you're like, wow, I really want to know more about 3D printing and paleontology, Aaron Johnson and myself wrote a paper about this in Frontiers in Ecology and Evolution, and we paid extra to make sure it's freely available to anybody who's curious. So with that, I will take questions.
So how long does it take you start to finish to kind of produce one of the models that you used in your work?
So pretty long. CT scans are a few hours. Segmenting manually, when I was really bad at it, took like a few days. So that's already a week we're looking at. And then a lot of experimenting with 3D printing. And I don't think that's what people say often is you have an STL, you have a printable file, but you had to experiment with the printer. The printer that we used has a nanometer refinement. So if there was a hair or a fluff off my shirt or just anything off just a little bit, the print would fail. So a lot of that was experimenting with the 3D printer itself. So a few weeks, at least 2 or 3 weeks.
Okay, and then once you had the models themselves built, I mean, how long did it take to do the testing period of it?
That's pretty fast. Designing the models and 3D printing the models are hard and long, but actually running the experiments in my case is half a day. That goes fast.
So how did you measure the amount of stiffness in the spine? What units did you use?
Yeah, so I have a machine called it's a torquometer, right? So it measures how hard I'm pulling on something, and then I filmed what I was pulling, and we could see with ImageJ the angular deflection. So how far something. So I had a video, and as I pulled, I could see the bone was moving, and then I measured that on my computer, and then I plotted degrees along X and the nanometers, so how much I pulled in Y. And that slope of the line is your stiffness.
Okay, and I mean, about how big were these critters that you were working on?
Oh yeah, so Diadectes is about 2 meters, Archeria is about 2 meters, and a meter is 3 feet. So you're looking at 2 animals that are 6 feet long. K-Cops was half a meter, so like foot and some change. So dogs, I guess. And then Diplocaulus was about a meter, so 3 feet long.
So what's the biggest part that you had to print during this process?
Oh, I have a vertebral column that's about 9 inches long, which sounds small until you have to print it. It's not that small anymore because a printing bed, so something you have to consider when you're 3D printing is the size of your printer bed, right? So if your printer bed is only 4 inches by 4 inches by 4 inches, right? Because your bed is tall, as long and tall, right? Long, depending on the size of your bed, right? Its length, its width, and how tall the printer is, you might have to do it in parts. So if your bed is small at 4 inches and you have a 9-inch print, all of a sudden it's a very big print.
And how long would it take to print something that big? I mean, we've done some of it here at the museum. So how long would it take to print something like that?
So those prints, because I use draft printing, was 2 hours, but I've worked on really complicated prints like ammonites. So these like curly Q cephalopods. We've 3D printed some of their elements, and those take upwards of about 15 hours because they're complicated.
So can you talk a little bit more about the kind of software that you use? Did you use those more focused on sculpting or more focused on engineering or design?
So I've used both. So a lot of my work was done in Fusion 360, which is a CAD, so computer aided design. Engineers use it for building parts. And when I needed a software really focused on 3D prints, because STLs are huge files. So an STL has a bunch of triangles describing the shape, and some of my vertebrae would have 65 million triangles. So to reduce this to a usable file type, I had to use something like Meshmixer, which was designed for 3D prints. Someone that I've worked with has used Blender, right, which is for more animation stuff. And I know some people that use Maya. So jack of all trades and master to none.
Can you talk about any further research you might be doing with the parts that you've made, or any additional work?
Yeah, so I'm currently a postdoc in the General Robotics Automating Sentiment Perception Lab. So I'm really interested on how the evolution of spinal function may tell us something about if we use spines in robots. So that's sort of my next step, is to get really good at understanding spines in the past so we can form maybe some spine usage in the future.
That sounds like fascinating work. Are you able to scale the output of the printer for different sized animals that are in the same genus?
Yeah, so that's what's really, really cool about using 3D printing is we can isolate variables. So let's say you had a question about T-Rex toes and Velociraptor toes and Deinonychus toes and Giganotosaurus toes. Well, 2 of those sets of feet are really close to each other in size, Giganotus and T-Rex, but Velociraptor's feet aren't as large as T-Rex's. So if your question is about feet, but you're worried about scale, you can scale down print so everything is the same size, which is super useful for scientists because we love isolating our vertebrae, isolating our variables.
All right, we don't have anything else popping up in the chat. So thank you very much, Dr. Asia Carter, for being here today. Great presentation. Thank you for helping us discover more about our world. I want to thank all of our guests for being here today. Please join us next week on Wednesday, July 21st at noon for Join the Expedition Live. This is presented by Dr. Jason Shine, the Executive Director of the Bighorn Basin Paleontological Institute. This will be a lunch break science presented live from a paleontology dig out west. This is going to be our first time doing something like this at Lunch Break Science, so it's very exciting. You will not want to miss this. Definitely do register for that. You can register for that at smv.org. Each talk is free to attend and is open to the first 300 registrants. Thank you again for joining us today. Until next week, stay safe and stay curious.